Okay, 15.3 is applications of genetic engineering. So, have you eaten any genetically modified food lately? Well, if you've eaten corn, potatoes, or soy products in any of your meals this week, chances are you're probably close to 100% that you've eaten these foods modified in some way by genetic engineering. So, can a genetic engineering benefit agriculture and industry? Well, ideally, genetic modification could lead to better, less expensive, and more nutritious food, as well as less harmful manufacturing processes. So almost everything we eat and much of what we wear come from living organisms. Researchers have used genetic engineering to try to improve the products we get from plants and animals. Genetic modification could lead to a better, less expensive, or more nutritious food, as well as less harmful manufacturing processes. So since their introduction in 1996, genetically modified plants have become an important component of our food supply. One genetic modification uses bacterial genes that produce a protein known as Bt toxin. This toxin is harmless to humans and most other animals, but the enzymes in the digestive systems of insects convert this enzyme to a form that kills the insects. So plants with the Bt gene do not have to be sprayed with pesticides. So in addition, they produce a higher yield of crops. Other useful genetic modifications include resistance to herbicides, which are chemicals that destroy weeds, and resistance to viral infections. So there's a chart in your book that shows soybeans, cotton, various kinds of corn, and it talks about the genetically engineered crops in the U.S. The modification traits shown in the graph include herbicide tolerance, which is labeled HT, and insect resistant, which is the large letter B and the small letter T that we were talking about just a minute ago. And some transgenic plants may soon produce foods that are resistant to rot and spoilage. Engineers are currently developing genetically modified plants that may produce plastics for the manufacturing industry. Transgenic animals are becoming more important to our food supply. About 30% of the milk in the U.S. markets comes from cows that have been injected with hormones that make the recombinant DNA techniques to increase milk production. So pigs can be genetically modified to produce more lean meat or high levels of healthy omega-3 acids. And using growth hormone genes, scientists can develop transgenic salmon that grow much more quickly than the wild salmon. Scientists in Canada combine spider genes into the cells of lactating goats. The goats begin to produce silk along with their milk. The silk can be extracted from the milk and woven into a thread that can be used to create a light, tough, and flexible material. Scientists are working to combine a gene for lysosome, an antibacterial protein found in human tears and breast milk, into the DNA of goats. Milk from these goats may help prevent infections in young children who drink it. Researchers hope that cloning will enable them to make copies of transgenic animals, which would increase the food supply and could help save endangered species. In 2008, the U.S. government approved the sale of meat and milk from cloned animals. Cloning technology could allow farmers to duplicate the best qualities of prize animals without the time and complications of traditional breeding techniques. So how can recombinant DNA technology improve human health? Well, today it is the source of some of the most important and exciting advances in the prevention and treatment of diseases. Golden rice is a genetically modified plant that contains increased amounts of provitamin A, also known as beta-carotene, a nutrient that is essential for human health. Two genes engineered in the rice genome help the grains produce and accumulate beta carotene. So provitamin A deficiencies produce serious medical problems, including infant blindness, and there's a hope that the provitamin A rich golden rice will help prevent these problems. Other scientists are developing transgenic plants and animals that produce human antibodies to fight disease. So in the future, transgenic animals may provide us with an ample supply of our own proteins. Several laboratories have engineered transgenic sheep and pigs that produce human proteins in their milk, making it easy to collect and refine the proteins. Many of these proteins can be used in disease prevention. So transgenic animals are often used as test subjects in medical research. They can simulate human diseases in which defective genes play a role. So scientists use models use models based on these simulations to follow the onset and progression of diseases and to construct tests of new drugs that may be useful for treatment. This approach has been used to develop models for disorders like Alzheimer's disease and arthritis. So recombinant DNA technology can be used to make important proteins that can prolong and even save human lives. For example, human growth hormone, which is used to treat patients suffering from pituitary dwarfism, is now widely available because it is a mass-produced by recombinant DNA. Other products now made in genetically engineered bacteria include insulin to treat diabetes, blood clotting factors for hemophiliacs, and potential cancer-fighting molecules such as interleukin-2 and interferon. Gene therapy is a process of changing a gene to treat a medical disease or disorder. And in gene therapy, an absent or a faulty gene is replaced by a normal working gene. And the process allows the body to make the protein or enzymes that it needs, which will eliminate the cause of the disorder. There are some pictures in your book that show exactly in treating diseases, examples of the gene therapy. Just talk through those a little bit. So to deliver therapeutic genes to target cells, researchers engineer a virus that cannot reproduce or cause harm. The DNA containing the therapeutic gene is inserted into the modified virus, and the patient's cells are then infected with the genetically engineered virus. In theory, no, I don't have the Rona. That's just my yearly allergies and asthma. In theory, the virus will insert the healthy gene into the target cell and correct the defect. So, gene therapy can be risky. In 1999, an 18-year-old Jesse Gelsinger volunteered for a gene therapy experiment designed to treat a genetic disorder of his liver. He suffered a massive reaction from the viruses used to carry genes into his liver cells, and he died a few days later. For gene therapy to become an accepted treatment, we need more reliable ways to insert working genes and to ensure that the DNA used in the therapy doesn't do any harm. So genetic testing can be used to determine if two prospective parents are carrying the allele for a genetic disorder such as cystic fibrosis. Because the cystic fibrosis allele has a slightly different DNA sequence from its normal counterpart, genetic tests can be labeled DNA probes that can detect and distinguish the complementary base sequences found in the disease-causing alleles. Some genetic tests search for changes in cutting sites of restriction enzymes, while others use the PCR to detect differences between the lengths of normal and abnormal alleles. Genetic tests are now available for diagnosing hundreds of different disorders. The same genes are not active in every cell. So by studying which genes are active and which are inactive in different cells, scientists can understand how the cells function normally and what happens when the genes don't work as they should. Scientists use DNA microarray technology to study hundreds or even thousands of genes at once to understand their activity levels. A DNA microarray is a glass slide or a silicon chip to which spots of single-stranded DNA have been tightly attached. Typically, each spot contains a different DNA fragment. Differently colored tags are used to label the source of the DNA. So, for example, to compare the genes in cancer cells with the genes in normal cells, the messenger RNA would first be isolated from both types of cells. And the enzymes are used to copy the messenger RNA base sequence into single-stranded DNA labeled with fluorescent colors, red for the cancer cell and green for the normal cell. Both samples of labeled DNA would be mixed together, and they would then compete to bind to the complementary DNA sequences already in the microarray. If the cancer cell produces more of a particular form of messenger RNA, then more red labeled molecules will bind at the spot for that gene, turning it red. Where the normal cell produces more messenger RNA for another gene, the spot will be green. And where there is no difference between the two cell types, the spot will be yellow because it contains both colors. So how is DNA used to identify individuals? Well, DNA fingerprinting analysis, sections of DNA that may have little or no function, but that vary widely from one individual to another. So no individual is exactly like any other genetically, except for identical twins, which share the same genome. Chromosomes contain many regions with repeated DNA sequences that do not code for proteins. These vary from person to person. One example would have 12 repeats between genes A and B, while the second one has 9 repeats between the same genes. So DNA fingerprinting can be used to identify individuals by analyzing these sections of DNA that may have little or no function, but that vary widely from one individual to another. So in DNA fingerprinting, restriction enzymes cut the small sample of human DNA into fragments containing genes and repeats. Note that the repeat fragments from these two samples are of different lengths. This is talking about a DNA fingerprint picture in your book. Next, the gel electrophoresis separates the restriction fragments by size. A DNA probe then detects the fragments that have highly variable regions, revealing a series of variously sized DNA bands. If enough combinations of enzymes and probes are used, the resulting pattern of bands can be distinguished statistically from that of any other individual in the world. So DNA samples can be obtained from blood, sperm, or tissue, even from a hair strand, if it has tissue at the root. So the precision and reliability of DNA fingerprinting has revolutionized forensics, the scientific study of crime scene evidence. DNA fingerprinting has helped solve crimes, convict criminals, and even overturn wrongful convictions. To date, DNA evidence has saved more than 110 wrongfully convicted prisoners from death. This book was published in 2010, so you know it's more than that now. DNA forensics is used in wildlife conservation as well. African elephants are a highly vulnerable species. Poachers who slaughter the animals mainly for their precious tusks have reduced their population dramatically. So to stop the ivory trade, African officials now use DNA fingerprinting to identify the herd from which black market ivory has been taken. And when genes are passed from a parent to a child, genetic recombination scrambles the molecular markers used for DNA fingerprinting. So ancestry can be difficult to trace. The Y chromosome, however, never undergoes crossing over, and only males carry it. Therefore, your Y chromosomes pass directly from father to son with few changes. Similarly, the small DNA molecules found in mitochondria are passed with very few changes from mother to child in the cytoplasm of the egg cell. Because the mitochondrial DNA is passed directly from mother to child, your mitochondrial DNA is the same as your mother's mitochondrial DNA, which is the same as her mother's mitochondrial DNA. This means that if two people have an exact match in their mitochondrial DNA, then there is a very good chance that they share a common maternal ancestor. Y chromosome analysis has helped researchers settle long-standing historical questions. One such question: did President Thomas Jefferson father the child of a slave? That may have been answered in 1998. DNA testing showed that descendants of the son of Sally Hemings, a slave of Jefferson's Virginia estate, carried his Y chromosome. This result suggests Jefferson was the child's father, although Thomas Jefferson Foundation continues to challenge that solution.